All right, welcome back. This is Mr. Finiseth, and we're again dealing with Chapter 8 and some of the chemical aspects of biology. Today's conversation is dealing with enzymes and how enzymes work to speed up metabolic reactions, and we're talking about energy barriers, activation energy. A lot of this stuff you should remember, hopefully, from chemistry, because we do spend a lot of time on this in the section on thermodynamics, which deal with chemical reactions being exothermic or endothermic. And we've talked about some of that stuff already. Our goal today then is to look at how we can take all these thousands of chemical reactions that are taking place inside cells on a regular basis and speed them up. If they were to react on their own, it would go very slowly, too slow for the cell to be able to survive. And so we're going to need some type of catalyst that is going to speed up the reaction without damaging what's going on in that chemical reaction. And with living things, we're talking specifically about proteins that are called enzymes. Enzymes are a group of catalysts that work in cells to take specific reactions, like say the hydrolysis of sucrose, and speed that up. If you had a beaker of sucrose and you wanted to wait for that to break down on its own at room temperature, it would take years for this chemical reaction to happen. The breaking of this bond would literally take years in a beaker of sucrose that's just by itself at room temperature. But if we add a pinch of our enzyme, sucrase, the ASE ending is an ending that we use whenever we're talking about an enzyme, and it is going to be specific to the reaction that it is tied to. Remember, enzymes are specific. Remember, you cannot take any enzyme and just put it in with any chemical reaction and get products out of it. They are very specific for the type of reaction that they're doing. So sucrase works with sucrose, and that chemical reaction involves the breaking of this chemical bond that you see here to give us glucose and fructose. Every chemical reaction between molecules involves breaking chemical bonds or forming chemical bonds, and this is going to require some energy. There's going to be a need for an initial energy input to get this reaction started. There's a little push that is going to be required, and that push is what we call activation energy. In chemistry, is being provided through getting out a Bunsen burner and heating up the beaker that has your chemical reaction going on in it. Well, the problem is with a living thing, you don't really want to strap a Bunsen burner to a living thing and heat them up, because that's just not going to work. Enzymes are very sensitive to temperature because they're proteins. They have a specific shape, and we have hydrogen bonds and ionic bonds that are taking this protein and folding it over so that it maintains that shape. So there's a lot of weak bonds. If you heat that molecule up, it's going to start to vibrate and move, and it's going to snap all those bonds, and that protein basically is going to fall apart. And if it loses its shape, it loses its ability to do its job. So again, this is a very common diagram that you will see in chemistry textbooks, and in this case, Chapter 8 in the biology textbook, where we have a series of reactants that we would like to combine together, and we would like to break a couple chemical bonds so that we can form a couple new chemical bonds and change our arrangement of atoms to give us some new products. The push that is needed to get this going, the breaking of bonds and the forming of new bonds, requires an input of activation energy. This hill that you oftentimes hear textbooks talking about, we have to climb this hill in order to get this reaction to roll downhill. But if we add an enzyme, we can then lower this barrier. And the way that it's lowered is the enzyme is helping align molecule A with molecule B to get the correct orientation so that the bonding can take place. Or maybe it's bringing the molecule into that active site, and it's starting to twist it and stretch it so that we start to break those chemical bonds more easily, and therefore the hill that we're climbing is significantly lower.
Exergonic reactions occur spontaneously. What keeps molecules from breaking apart and cell chemistry from racing out of control? For any reaction to occur, even a downhill reaction, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, E sub A. This required energy input represents a barrier that prevents even energy-releasing exergonic reactions from occurring without some added energy. How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules, referred to as the enzyme substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction.
All right, so that animation did really kind of a nice job of walking you through the whole thing. And again, one of the key points is that our enzyme that we're working with is very specific. You cannot take any enzyme to run a particular chemical reaction. That enzyme is going to have a distinct shape to it, like you saw on the animation, and there's some substrate that will fit into this active site. Those are two terms that you definitely need to know for section 8.4, the term substrate and the term active site. The shape is the active site that our substrate is going to fit into, and again, this is going to help bring things together or help pull things apart, which then in turn lowers the amount of energy that is required to get the reaction to take place. And if it requires less energy to bring this stuff together or take this stuff apart, then naturally the reaction should flow to the products much quicker than if it did not have the enzyme assisting with this process. So again, I think that animation was a pretty good one to go through. Here's your terms again. If you're looking at definitions, the substrate is the molecule that the enzyme is acting upon. Once that substrate gets plugged into the active site, it becomes the enzyme substrate complex, and you would find that at kind of the peak of the hill. That complex is temporarily arranged in such a way where now it can either pull something apart or bring something together, and it's just there for a fraction of a second, and then boom, it does its thing and moves on to grab another substrate. And again, that's kind of a key piece to this whole deal. One enzyme is doing thousands of the same reaction. It's going to grab a substrate, act on it, dump it. Grab another substrate, act on it, dump it. And just keep going until it runs out of substrate. I mean, really, that's what it comes down to. Or we have some inhibitors that are going to come in, and we'll talk about that a little bit later in the chapter, and how they slow the work of an enzyme down by interfering with the active site, and how substrates would fit into that chemical. We call that an induced fit, where the chemical groups or the active sites into positions that enhance their ability to catalyze the reaction. It's going to bring that chemical in and allow it to orientate itself in such a way where the reaction can take place. Here is a diagram of a very complex enzyme, and you can see this massive protein molecule has a distinct shape, and it's designed this way to give us an active site that will only work for a particular substrate, whatever it is. It could be multiple reactants that are coming in, or it could be one reactant that is coming into this active site. Once it's in the active site, then you can see that the protein may fold in such a way to bring those molecules together, or it may twist to pull those molecules apart. And so we're talking about a number of different things that an enzyme can do. We talked about the orientating the substances correctly. We talked about straining chemical bonds so that they want to start snapping. We haven't really talked about a favorable microenvironment, but we'll do that next. And then covalently bonding to the substrate, we'll talk about that a little bit as well here in the next slide. So here we see a couple substrates that we want to fit into this enzyme, and if we were working without the enzyme, we would be relying on these two to just kind of bump into one another and be correctly orientated so that they bond together and we have our complex. Well, random chance really isn't going to work very efficiently in bringing these substrates together. But you can see that this substrate is only going to fit into one part of the enzyme. It will not fit down here. Same thing with this other substrate. By getting them to be shape specific, we can get them lined up in such a way that we can start to bond them together and create the product that we're looking for. In this case, we've kind of modified both of these molecules into something new. That enzyme will only work for those substrates, and so if it doesn't have anything to do, it just sits and waits. Local conditions, the microenvironment that they were talking about on the previous slide. What do we mean by that? Well, again, temperature, pH, those are things that you can't always change in the cell. We don't want to make the cell really acidic in order to speed up a chemical reaction, or raise the temperature to speed up a chemical reaction, because it'll be very damaging to the cell. But enzymes, they can have pockets. Their active sites could be made up of amino acids that are acidic in nature. And therefore, this little pocket within the cytoplasm of the cell is kind of this own mini environment that is very low pH, and that maybe is the type of situation that you need, because we need to attach hydrogens to some substrate, and that's what acidic environments entail, is lots of hydrogens. And so now we've created this little pocket where the chemical reaction can take place because we have a whole bunch of hydrogens waiting to be used. Optimal temperatures, when we did the AP lab, of course, some of the individual research was done with pH as well as with temperature, and one of the big things that you should have seen is that an enzyme often functions at the temperature at which the organism is functioning. For example, we're warm-blooded animals, and our body temperature is fairly warm, 98.7 degrees, and as it turns out, that is the optimal temperature for the enzymes that we have. They like to work at that temperature. They work very efficiently at that temperature, and if we lower our body temperature, raise our body temperature, we start to run into some real problems with those enzymes, and it could be fatal. If you're talking about a cold-blooded animal, like a reptile or an amphibian, they have a whole different set of enzymes that are going to work at temperatures much lower, because of the fact that reptiles and amphibians are oftentimes operating at temperatures, body temperatures, that are much lower. We also have a need at times to slow chemical reactions down. It's really important that we don't have chemical reactions just blow out a particular substrate. Because if it's an exothermic reaction where you're talking about the liberation of heat and energy very rapidly, that could be very destructive to the cell. So we have a need to kind of slow down some of these enzymes once in a while. We have competitive inhibitors and we have non-competitive inhibitors, and that's what we're going to mention here real quick. Yeah. And there's some examples here on this screen. This would be a normal binding situation where our active site and our substrate fit nicely together. But if some inhibitor comes lumbering in here and attaches itself so that the substrate can't get in, you've basically put that enzyme out of commission. Same thing here, this non-competitive inhibitor is attaching somewhere else in the enzyme, distorting this shape so that now this really doesn't fit in that spot anymore. And again, we've made that enzyme we've made it useless. Basically, it can't do its job.